Okay, so the class today will be actually the first bioinformatics class that we will have. Until now we were obtaining insights into the details of R. So we were discussing the different data structures that we have in R. But today we will go to the position weight matrices, which is really a bioinformatics topic. Okay, in order to understand what a position weight matrix is, in short it's called PWM, we have to go through some biology. So all this that I will describe today is a simplistic version of the reality, but nevertheless it captures several aspects of what is happening in a cell. So this is the DNA, again as a straight line, and at some location of the DNA there is a gene. So this is a gene. Now this gene will be transcribed and it will generate some mRNA here. So the more it will be transcribed, the more mRNA it will generate. Now the initial position where the transcription starts is called TSS, or transcription start site. Now a basic question in molecular biology is how much DNA will be produced, how much RNA, will be produced, and when the production of mRNA will start, or when transcription will start. So for these two questions, in order the cell to give a satisfactory answer to these two questions, it must have a regulatory mechanism that it will regulate transcription. And a player in this regulatory machinery is the transcription factor. So a transcription factor is a protein, this protein A for example, that can bind to DNA. So here you have a binding between DNA and this protein, and when this protein is bound to the DNA, usually before the TSS, before the transcription start site, then transcription of the gene may start and mRNA is produced. So the regulation of the gene transcription is achieved by the protein A here, the transcription factor. Now this protein has a property to bind to the DNA. However, it does not bind anywhere in the DNA. It binds only to specific sequences. So in this example, this protein can bind to a sequence ACCTAAACT. However, this is not the unique sequence that this protein may bind. It can bind to this sequence and to other sequences that are similar to this one. For example, in a sequence that it will be ACGTAAACT. There is no certain rule that will define which sequences are appropriate for this protein to bind. However, we have observed that this protein may bind to similar sequences, that all the sequences that this protein can bind to is similar to each other. Other proteins, for example a protein B, another one, binds to a different sequence of DNA. So each transcription factor has its own preferences. Now our task is to describe all the plausible locations that the transcription factor may bind and assign a score in these locations. In a sense that the higher the score, the more plausible is that the transcription factor will bind there. And since this set of DNA sequences that this protein can bind to is not, they don't match 100% to each other, but some differences are allowed, we say that this protein can bind to a motif, not to a sequence, but to a motif, because a motif incorporates several different sequences that matches to each other. You will see that in a minute what I mean. So let's have an example. Let's say that for this protein A, we have the following DNA sequences that on which this protein can bind. So this protein A can bind to a sequence CCTAAACT, but also to the CCTTAACT. Well, this is just an artificial example, so I'm not sure that there is such a motif as this one, but it's an example that reflects somehow what is happening in reality. So these 5 DNA sequences have been found experimentally that this protein A, in which this protein A can bind. Okay, so all this set comprises a motif, and we're going to describe this motif, and this is our task 1, and the task 2 is to use this information in order to discover other sequences that may belong to this motif. So other similar sequences to this one that exist in the DNA and may bind to this motif, may be a part of this motif. Okay, let's see what we will do. To answer the first task, this one, which is to describe this motif in some compressed way, we have the notion of the position weight matrix. Okay, so again, let's have these different sequences that this protein can bind to, and for our class, so for this lecture here, we assume that all the sequences have exactly the same length. Now here the length is 1, 2, 3, 4, 5, 6, 7, 8. So the sequence consists of 8 nucleotides. Okay, and let's say that this is cell. Now, if we have a closer look to these sequences, we see that it doesn't seem that all positions are exactly equivalent. For example, in position A, in position 1, both C and G is allowed, of course with different frequencies each one, so it seems that C is more frequent than G. In position 2 only C is allowed, position 3 only T is allowed, and so on. So for some positions it seems that we should be very strict and allow only one nucleotide. For other positions we don't need to be so strict, more than one nucleotide is allowed, but again with different frequencies. So there are preferences. But even in this position, in the position 2, that in our sample of DNA sequences we have only one letter, it might be that some other letter is allowed, but we haven't found it in our sample. So we must be a little bit more flexible here, but still we must give a high weight, let's say, you will see that later on, in C instead of any other letter. Okay, so these are some observations, and now we want to somehow represent this in another way. Okay, what we are doing, I will describe here the algorithm and then we can discuss it. So the first step is to create a 4 by L matrix. So a matrix with 4 rows and L columns. Remember that L is the length of each DNA sequence of each DNA sequence that a protein can bind to. And we initialize this matrix with zeros. So this is the initialization and the construction step of the matrix. And then we go back to the sequences, so this set here, and we count for each position, so for the first position, for the second position, for the third, up to the 8th position, how many ACGTs we find. So in the first position there are 0 A's, and we write 0 here. And then in the first position we have 1, 2, 3, 4 C's, we write a 4 here, 2 G's and 0 T's. So we fill the first column of the matrix with the information from the first column, from the first position of the sequences. Okay, that's good. Then we go to the second position, and here it's clear that everything is C, so 6 for C and 0 everywhere else. We go to the third position, to the 4, to the 5th, to the 6th, to the 7th and to the 8th position, and we do the same thing. And this matrix is a count matrix, so it just counts how many nucleotides we have at each position. Again, the dimensions of this matrix is 4 rows, and the number of columns is the same as the length of the motif sequence. Now the next step is to convert this count matrix to frequencies. This is a very simple step because we just divide each number with the sum of the column. So the sum of this column is 6, we divide everything by 6. In fact, everything is 6 here, so we could just divide everything by 6, but we should not do that. Let's imagine that here some information is missing, so instead of A we have the letter N. Now N means missing for DNA. Missing means that we don't know if it is A, C, G or T. We don't know which of the 4 letters is, and we just put an N here. If this is the case here, then for this column we will not have 6 nucleotides, so we will not have a 6 here, but instead we will have a 5 here. Okay, just to make this clear why we need to find the sum of every column and divide with the sum of every column, because it's possible that some information will be missing. Okay, we have now the frequencies, and this matrix is called position probability matrix, or PPM. And we observe that we have several zeros here. So a 0 means that in some specific position we didn't find a nucleotide. For example, in this position here, in the 7th position, we didn't find any A or any T in our sample, so we put a 0. Now we don't like zeros, as you will see later on. I will explain that later. So we add what is called a pseudo count. So a pseudo count is just a small positive number in order to avoid to have completely zeros here. So an example is 10 to the minus 6, 10 to the minus 4 is another option, 10 to the minus 8. It doesn't really matter so much, just a small number. So then we will have here 0 plus 10 to the minus 6 4/6 plus 10 to the minus 6 and so on and so forth. We will do that for every cell here. So after this operation we will have no zeros. Okay, and now we do the following step, and this is a bit tricky. This is the frequencies of the ACGT that we observe at each position of the DNA sequences that our protein can bind to. What we need then is what we would expect by chance to find in every position of the sequence. So there are two trivial answers to that. The first answer is that all letters, all ACGTs, have exactly the same frequency, so by chance I would expect just 1/4 to be A, 1/4 to be C, 1/4 to be G, and 1/4 to be T. So 0.25 probability for each nucleotide just by chance. But if we go to the initial DNA, and I will explain the second solution, then it might be that all this DNA here does not have the frequency of A equal to the frequency of C equal to the frequency of G and equal to the frequency of T. It might be different, the different frequencies. They will sum up to 1, but we have no reason to believe that these 4 nucleotides are found in general on this DNA in the same frequency. So remember that this DNA is quite long. It can be million of base pairs, or 1,000 millions, whatever, of base pairs, bases, and we don't have any reason to believe that these frequencies could be exactly the same, and they will be 0.25. Which means that if we would expect something by chance here, then these will be similar to the frequencies that we would observe elsewhere in the genome. So for example, if in this genome the frequency of A will be 0.3, the frequency of C will be 0.35, the frequency of G will be 0.25, and the frequency of T will be 0.42, so all this sum to 1, then also here we would expect to observe just by chance these frequencies of ACGT. Either the first solution that we put everywhere 0.25, or we count the frequencies for the whole genome and we assume that this is what we expect by chance. We need, and we take somehow what we call the frequencies of ACGT that we expect by chance. Okay, so these are the two trivial solutions, and what we do then is that we divide each frequency with what we observe, with what we expect just by chance. So here we get this fraction, which is 0.6 that we have for this position, plus epsilon is a pseudo count, divided by 0.25. Okay, now this will be a small number because this is 0, and this will be epsilon by 0.25. But if we go to another one, this 4/6 for example, it will be 4/6 plus epsilon divided by 0.25. And this number approximately tells us how much more frequent, how many times more frequent is C in this position, in the position number 6. So this 6, 7, 8. How much more frequent is C in this position compared to what we expect by chance? If it is greater than 0.25, it means that we expect to see a C more frequently than what we would observe just by chance. Okay, and after we take this ratio here, we find the log of this ratio. So this number, this number inside the log is a number that is greater than 1. If it is X's number, is greater than 1, is greater than 0, and it is it can be greater than 1, but in this case where we put 0.25, it can go up to 1 divided by 0.25, which is 4. 1 plus epsilon is approximately 4. So the logarithm of this value is either a very negative number, because everything that's smaller than 1 will be negative, or some maximum value, which could be the log of this value. So we find the log of each cell here, and this is the position weight matrix. Okay, let's see some notes on that. Now, if the frequency of a nucleotide X at position I is higher than what is expected by chance, then the position weight matrix of this nucleotide at position I is greater than 0. Otherwise, it's less or equal to 0. What I mean by that is that if what we expect by chance is 0.25 and we find a frequency which is greater than this 0.25, let's say 0.5, let's neglect epsilon for the moment, the log of this number, since this number is greater than 1, will be positive. Okay, and otherwise this will be negative. So if it is less than what we expect by chance, it will be negative. So the higher the frequency, the higher the value that we will have for this nucleotide in the position weight matrix. There is this maximum value, as I explained before, and there is a minimum value, which is also this one. It will be a quite negative number. And now you understand why pseudo-cows are needed, because if we would have 0 here, then we would have to calculate the logarithm of 0, which is minus infinity. We don't want that, so we put an epsilon here. And another subtle thing on the position weight matrix is that we assume that the different positions on the motif are independent, which means that if we're going to calculate the probability to observe a certain motif given the position weight matrix, this will be just multiply the probability to observe NA at position 1. So this is not an index, it's not a power, it's just A at position 1 given the position weight matrix, multiplied by the probability to see a C at position 2 given the position weight matrix, and so on. Now this is if we have the probabilities. However, you remember that from this step we pass from probabilities or frequencies to logarithms, and therefore instead of finding the probability to see a motif, we can score a motif, a sequence here. We can score a sequence by finding the sums of the respective position weight matrix values, and this is associated somehow to the probability of finding a sequence given the position weight matrix, because if we multiply probabilities, it's similar as if we sum the logs of this number. Now this is not exactly the probabilities, because we have divided by the what we expect by chance, but it's somehow it is related to this probability. So the higher the score we obtain here, the more a sequence matches to the position weight matrix, or the higher is the probability to observe a given DNA sequence for this position weight matrix. So I have described here the algorithm how to generate position weight matrices. I have explained what they are. They really capture a lot of information of the DNA sequences that a protein can bind to by compressing all the information that is found in different DNA sequences in a way, in a matrix that comprises 4 rows and some columns here. And we will see in the next class how we can use this information to score all the possible positions in a sequence and see whether it can be a location that a protein can bind or not. That's for today. Thanks a lot.